title of today's webinar is Advanced 3D Cell Models for Cancer Using Real-Time Live Cell Analysis. So we know that 3D in vitro models have been used in cancer research as an intermediate model between in vitro cancer cell lines and in vivo tumours. So in this webinar, I hope to describe a range of 3D culture models and cancer-related applications, show how real-time live cell analysis and these 3D cancer models can provide additional insight when capturing biological parameters such as morphology, growth and viability, and how the IncuCyte's integrated image acquisition and analysis is really ideally suited to these very 3D cultures. And finally, how more complex co-culture models and organoid cultures are compatible with the IncuCyte. So before doing so, I'd like to first revisit the known hallmarks of cancer as first described by Hanahan. So these are highlighted here and basically describe a set of underlying attributes which cancer cells display. You can see here a suite of 2D live cell analysis assays and techniques which have been used to understand and assess key phenotypes as well as underlying mechanisms associated with these hallmarks. Now here are those very same applications, only now using 3D cultures, which one could possibly argue align more with the in vitro setting and have potentially more translational opportunity. Now, with a drive to more closely align cellular models used in research and early phase drug discovery with the preclinical models and patient tumours, being mindful of the tumour microenvironment is key. We know how the tumour microenvironment is complex and not only includes tumour cells, but other cell types and an extracellular matrix. So iterations of advanced cell models, which strive to really recapitulate the tumour microenvironment, as I said, are key to understanding the influence it has on all of the hallmarks of cancer. So here is a flavour of the 3D models and related oncology applications that will be described within this webinar, including single spheroids, multispheroids, both in monoculture or co-culture, in the presence or absence of an extracellular matrix, and the culturing of organoids embedded in Matrigel domes. So I just want to spend a few minutes describing those 3D models that will be referred to in this webinar. These include scaffold-free and scaffold-based models. Firstly, this schematic shows single spheroids created in the absence of a scaffold in either 96 or 384 well round bottom ultra low attachment plates. So we know that these protocols allow for the production of very uniform size controlled single spheroids, where you create a single spheroid in each well that really takes on the spatial or zonal regions characteristic of solid tumours, as well as the physiochemical environment with respect to nutrients, waste and oxygen gradients. And these models really tend to avoid the use of complex biomatrices. Here is a scaffold-based model of multispheroids formed on a base of Matrigel in a 96 well flat bottom plate. So it's the lack of standard spheroid size in this model, as well as morphology, that's very characteristic. Spheroids are much smaller. They form and grow by natural aggregation on or within an extracellular matrix that closely mimics the tumour microenvironment. And this final model here describes organoids cultured in Matrigel domes within 24 or 48 well flat bottom plates. And unlike the previous two models, this one describes structures which are very much distributed in different planes within the extracellular matrix. Now, all of these protocols have been validated for use on the IncuCyte platform, and coupled with integrated software specially designed for these 3D cultures, allows for the continuous live cell monitoring and quantification of biological parameters that I will describe throughout the webinar. So here what you're looking at are some example videos of single and multispheroids created using the first two protocols I described. You can see that the IncuCyte is able to capture both high definition phase and bright field images Now, some of you may be very familiar with the challenges of consistently segmenting or masking phase images, in particular imaging of single spheroids of larger than 500 microns in diameter, where the center of the object often appears out of focus, or poor or out of focus phase images of multispheroids growing on the meniscus of an extracellular matrix gel. So to circumvent these challenges, we've developed a proprietary method to capture depth of focus bright field images, referred to as DFBF, for both of these models. Note how the IncuSight is able to consistently segment these bright field images over time, as shown by the yellow outline mask. Now this slide illustrates how the size or area of a population of multispheroids can be tracked and quantified over time using a label-free approach. You can see again that the IncuSight is able to achieve this for multispheroids which have distinctly different morphologies, round as illustrated by the MCFs on the top panel, and a more branched stellate morphology adopted by the MDA cells, which is very characteristic of an invasive phenotype. The graphs show the different temporal growth profiles of the two cell types, tracking not only the growth, but the lack or potentially the death induced by the treatment of a well-known cytotoxic agent, camptothecin. So just returning for a few moments to how assessment of images and videos through time can potentially provide additional biological insight. So what I'm showing here is some validation data of how the size of A549 multispheroids is dependent on cell seeding density. If you just focus on the left-hand plot, you can see that cells seeded at the higher density achieve a growth plateau sooner than cells seeded at the lower densities. And this is something you may well predict and reasonably speculate is due to the depletion of nutrients over the time frame of this culture, which is 10 days, up to 10 days, sorry. The middle plot quantifies the number of multispheroids that are formed across each seeding density and how that number changes with respect to time. So interestingly, spheroid count decreases for the highest seeding density, but remains relatively constant for the lowest. Closer inspection of the videos and images show that this is due to neighboring multispheroids merging with one another to form larger single spheroids. If you look at the average size of the multispheroids on the right-hand plot, you can see that cells seeded at all three densities have the same growth rate over the first 72 hours post-formation, with cells seeded at the highest density being the smallest. And since they are further apart from one another, the lower density multispheroids are less likely to merge and also have more room to grow to a larger average size. So you can use this type of matrix or validation experiment described and these metrics, which will ultimately allow you to define optimal culture conditions for your assay. If, for example, your objective is to avoid or minimize merging of multispheroids in your experiment. So earlier I described how bright field image capture from control wells and treated wells with a known cytotoxic agent, camptothecin, could be used to quantify growth and death of multispheroids. Now you could possibly argue that this is not actually death but a lack of growth we are seeing in these wells. And one way to verify death would be to use a cell health reagent such as IncuSight Cytotox Green. So this is non-perturbing dye to normal healthy cells with a very low intrinsic fluorescence, and only becomes fluorescent upon binding to DNA in dying or unhealthy cells where the membrane of the cell integrity is somewhat lost. So here we have a 10-day experiment looking at the cytotoxic effects of camptothecin on scaffold-free spheroids over time, and the assay is performed in the presence of this Cytotox Green reagent. So looking at the bright field videos, you can see that the control spheroid continues to grow while the treated one doesn't. Its 3D structure also appears to break down, and you can see an accumulation of debris around the periphery of that object. The area of both of these spheroids is shown in the time course plot and aligns with what you see in the videos. The fluorescence videos show a strong fluorescence signal in the treated, smaller spheroid and a much dimmer signal in the larger control one, which reveals itself much later in the time course. There is also appears to be an overlap between the bright field and fluorescent images with respect to the debris, suggesting that we are clearly looking at dead cells here. And quantification of this cytotoxic mode of death is determined without the need to perform fluorescent object masking, which can be fraught with regard to it being very subjective, and where interpretation of the results is very much influenced by the threshold set by the user around the fluorescent segmentation. Instead, the IncuSite is able to return the intensity of the fluorescent signal within the boundary of the bright field object. That is a fluorescent signal with respect to the size of the spheroid. And this is verified by the bottom plot on the right-hand side, showing an increase in the intensity in the green signal in the much smaller treated wells which are dying. So the IncuSite cell health reagent can be extended and used with multispheroids. Here we have MDA cells which are treated again with camptothecin. As with the single spheroids, quantification of the bright field images in the top panel illustrates the continued growth in the control wells, so as seen by the teal curve on the plot, and the lack or minimal growth tracked in black of treated multispheroids. The use of annexin 5 in this example and quantification of the green fluorescence intensity within the boundary of the bright field objects in these images illustrates apoptotic death by camptothecin. Now, these dual readouts can be further multiplexed with an alternative measure of spheroid growth and viability. So these cells also express a red fluorescent protein. Now, you can see in the bottom panel of videos an increase in the intensity of the red fluorescent signal in growing spheroids and a decline observed in the treated spheroids. The corresponding time courses demonstrates how the fluorescent signal from stably integrated fluorescent proteins can be used as a surrogate for growth and viability. And here to illustrate how you can perform robust pharmacological analysis using a 3D multispheroid culture in the presence of an extracellular matrix. We have MCF7 cells treated with 3 compounds, and you're looking at the IncuSite deep views of the phase and now the bright field images. As I said, these cells express a red fluorescent protein, and it's these visualizations that allow you to very rapidly assess the treatment effects on multispheroid growth and viability. And the plate views here show the size and fluorescence intensity quantification of those images, returning efficacy values for inhibition of spheroid growth, so that's looking at inhibition of the area of the object, and inhibition of viability. So you're looking at the red fluorescent intensity metric as I described within the boundary of the bright field object. So I'd now like to focus on another application, which is the study of solid tumor invasion. I already described earlier how cell invasion is considered as a notable hallmark or phenotype of malignant cancers. And we know that the ability of tumor cells to form a metastatic or spreading tumor is largely determined by the cell's ability to reorganize its cellular morphology and degrade the extracellular matrix. So the IncuSite invasion protocol is shown at the top here in a schematic form, where single spheroids are formed in 96 well round bottom ultra low attachment plates, followed by the addition of Matrigel. In combination with the IncuSite allows you to study the invasive capacity of malignant tumor cells. Again, this enhanced depth of focus bright field image acquisition enables real-time kinetic imaging of the 3D single spheroids, which are now, as I said, embedded in an extracellular matrix. The example here is of U87 spheroids, and hopefully you can see the characteristic stellate invadopodia developing in the control wells, which is inhibited by cytokinase D. The metastatic potential of 3 tumor cell lines shown here was assessed using the protocol I've just walked you through. The videos show high contrast DFBF bright images, which are automatically quantified with respect to 2 size metrics. The first is the area of the whole spheroid, which is shown by the yellow outline mask on the panel of the top videos, and the area of the invading cells. And it's this area which quantifies the metastatic potential of spheroids and is shown in the blue outline mask on the bottom videos. You can see that the IncuSite is able to accurately differentiate the invading cell area from the spheroid body across the 3 cell types. And these kinetic profiles tracking total area and invading area are shown on the plots. And although different in overall size, I beg your pardon, so you can clearly see from those plots, the U87 spheroids showed the greatest invasive potential. You can see that its area increased to approximately 2-fold that of the HG1080 cells and approximately 4-fold that of the size of the A172 spheroids. And what's of note here is that although different in overall size, with the HG1080s having a larger total area, and you can see that in the left-hand plot, the A172s and the HG1080 spheroids showed comparable invasive potential. So here I'm looking at the right-hand plot. So this difference in invasive potential across the 3 cell types may indicate very distinctive mechanisms of cell motility and invasive modalities. And I think you would need to perform some additional experiments to fully understand the mechanisms that are at play here. So we're very aware that metastasis is a multi-step process, as I said, in which the extracellular matrix and cancer cell cytoskeleton interactions are very much essential to promoting the invasive likelihood of tumor cells. And here what we're showing is the relationship between the extracellular matrix properties, for example, its rigidity, the invasive potential, and invasive phenotype of HG1080 spheroids, which have been embedded in a range of Matrigel concentrations. The DFBF images and quantification of whole spheroid area show the dependency of spheroid invasive potential on Matrigel concentration. So the images also illustrate the effect of Matrigel concentration on the density of the spindle-like protrusions known as invadopodia. And you can see that there's marked density and elongation of this invadopodia when using the highest concentration of Matrigel in this experiment. So now looking across 3 cell types, we can observe a bell-shaped relationship between the ECM concentration and invasive potential of U87s and the A172 spheroids, with the greatest invading cell area attained at the 2.5 or 2.25 mg per mil test concentration. This is in alignment with reports which suggest that an optimal ECM concentration cells at the periphery of the spheroid very much align with the matrix fibers and rapidly invade the surrounding ECMs. Now, we would need to, of course, firm up this observation by performing a much wider test concentration range for Matrigel. Now, ultimately being able to separate treatment effects on the inhibition of spheroid invasion versus inhibition of spheroid proliferation is key to defining the mechanism of action of your treatment. Here we illustrate how this spheroid invasion application and associated quantification and analyses is able to do this. So we have U87 spheroids which have been treated with cytokinesin D, an inhibitor of actin polymerization, and the PP242 compound, an mTOR1/2 inhibitor. If you just focus on the DFBF images, you can see that they reveal inhibitory effects of both of these compounds on spheroid size. And this is reported by the reduction in the whole spheroid area. So I'm looking at the black bars in the bottom left-hand panel, as well as the invading cell area. So those are the turquoise bars. Now you can see that the inhibition by cytokinesin D is more marked with respect to the invading cell area. Now, although similar in overall size at 96 hours post-treatment, we can see that for cytokinesin D treated spheroids, only 30% of its overall size comes from the invading cell region, while this is almost 80% for the PP242 treated spheroids. So this is very much confirming that cytokinesin D is inhibiting the invasive phenotype in these spheroids. Now, we can see that the spheroid body of PP242 spheroids is much smaller with respect to the control. So I'm now looking at the images again. The size of this spheroid body can be derived by subtracting the invading cell area from the whole spheroid area, and thereby providing a means of assessing the effects of your treatment on spheroid proliferation. So you can see on the bottom right-hand bar graph there, the notable effect or inhibitory effect that the PP242 compound has on proliferation. So antibody-dependent cell cytotoxicity assays using 3D spheroid models are increasingly being used to develop improved immunotherapies. And here is an example of an ADCC assay looking at the growth and immune-targeted destruction of 3D single spheroids. The top two videos show how a red fluorescent protein expressed in SCOF spheroids treated with or without Herceptin continue to grow, and you see them increase in size and in fluorescence intensity. Minimal death, that is, the loss of fluorescent signal, occurs upon spheroid treatment with non-activated PBMCs. So that's the bottom left-hand video. In contrast, Herceptin-activated PBMCs displayed enhanced and targeted killing of the spheroid, which is manifested as a loss of fluorescent signal and lack of spheroid growth. So Herceptin-activated PBMCs cause a concentration-dependent death of SCOF spheroids, as shown in the time course plot, where spheroid proliferation and death quantified using fluorescence intensity within the bright field boundary metric I spoke of earlier. Note that targeting the T cell population with a cocktail of anti-CD3 and IL-2 resulted in maximal death of the spheroid. Now we can extend this application to multispheroids, which form in the presence of an extracellular matrix. So Herceptin-induced ADCC was evaluated now across 3 cell types stably expressing either a red fluorescent protein or a green fluorescent protein. The images and time course plots show specific killing of HER2 positive, those being the SCOF3 and the BT474 multispheroids, but not HER2 negative multispheroids. So I'm looking at the MCF7 cells. And we can see that Herceptin induced a concentration-dependent cytotoxic effect in the HER2 positive cells, but not, as I said, the HER2 negative. And Herceptin also showed sensitivity differences across the HER2 positive cells. So you can see that in the CRC plot with the differences in IC50s across SCOF3s and BT474s. So as I highlighted earlier in this presentation, in vitro models that go somewhat to recapitulate the tumor microenvironment via incorporation of an extracellular matrix and additional cell types such as stromal, endothelial, or immune cells allow you to assess your treatment effects, study chemoresistance, and immune tumor cell interactions. Here we're monitoring the effects of stromal cells, those being normal human dermal fibroblasts, on tumor multispheroid morphology. From the videos, you can see that the SKBR3 cells only form compact spheroids when co-cultured with fibroblasts. The kinetic quantification of this qualitative assessment, tracking spheroid size, is shown in the time course. Now, looking at the MDAs, they take on a more stellate morphology in the presence of fibroblasts. And interestingly, this appears to be revealed in a time-dependent manner, where multispheroids transition from this stellate branched appearance, which is evident at early time points, before returning to a more clustered, rounded appearance at later time points. So this is beyond day 6, which you're looking at in this panel of still images. And these stellate protrusions, as I said, were only observed 3 days post-formation in the monoculture setting. The bar charts show the total bright field area of spheroids in monoculture and co-culture and illustrate the impact of the fibroblasts on the multispheroid tumour size over time. So here we show how a 3D co-culture model is amenable to pharmacological profiling of compounds. A panel of 4 commonly used breast cell lines were co-cultured with fibroblasts and multispheroids allowed to form for 3 days prior to treatment with standard of care and cytotoxic agents, lapitinib, ZK164015 and camptothecin. We can see that inhibition of multispheroid growth caused by lapitinib and the ZK compound aligned with the known expression profiles of receptors which are targeted by these agents. So that is the dual EGRF and HER2 tyrosine kinase inhibitor lapitinib caused a concentration-dependent inhibition of SKBR3 and BT474 growth, while the estrogen receptor antagonist, the ZK compound, was a potent inhibitor of MCF7 growth and had very little, if any, effect on the multispheroids which were devoid of estrogen receptor expression. And the broad spectrum anti-cancer agent camptothecin, as you would expect, was equipotent across all 3 cell types. So having demonstrated the IncuCyte's ability to acquire and analyze images from 3D multispheroid co-cultures, you may want to consider using fluorescently labeled endothelial cells instead of fibroblasts, or possibly studying the impact of immune cells or stromal cells by creating a triculture. So the remaining section of this webinar focuses on a 3D organoid model where organoids are embedded within Matrigel domes. This schematic shows how cultures can be prepared in either 24 or 48 well microtiter plates with organoids, as I said, embedded in these domes. So using a proprietary approach to image capture, the IncuCyte is able to consistently acquire bright field images of organoids embedded in different Z-planes within these Matrigel domes. Note that the whole dome is being imaged here and all of the objects within are in focus. You can clearly see the budding crypt-like domains from the form around the lumen of the intestinal organoids, which mature around day 4. Also shown are examples of more rounded organoids with the pancreatic and hepatic cultures. Note that these cultures not only increase in size over time, but you may catch a glimpse of some of them bursting as the culture reaches maturity. Now, we can go on to quantify the differentiation and growth of the organoids as they remain disturbed within your incubator. The yellow outline mask in the videos shows the segmentation of the 2 distinctly different organoid morphologies I just highlighted. And the corresponding time course profiles track the size or area of the total population within the dome. And note the very different growth kinetics for both of the cell types. So defining optimal culture conditions and regimes is not only key for maintaining or expanding or cryopreserving your culture, but also for ensuring that cultures are consistently prepared for use in downstream assays. Integrated size and count metrics enables you to objectively define such conditions. Here we see for hepatic organoids, size and number is dependent on seeding density. Cultures seeded too high a density result in overcrowding, and beyond 144 hours on the plot, the average size of the organoids decline. Intestinal organoids, you can see here, take much longer to mature when compared to the hepatic organoids. And we can see seeding at too low a density, so just drawing your eye to the 1 in 12 split, prolongs this. In addition, organoids in these wells have a much smaller average size compared to cultures seeded at the higher 2 densities. The passage frequency of cultures often relies on an expert eye of a limited number of experienced users accurately documenting their observations. Now, making this a more objective approach avoids the need for extensive training and ensures consistent, unbiased operator-dependent regimes. Now, organoid passaging frequency is driven by indicators of organoid maturation, such as size, as I described on the previous slides, budding, and accumulation of debris within a lumen. You can kinetically quantify these indicators using the IncuSight's eccentricity or darkness metrics. So eccentricity tracks the degree of roundness of the organoids over time, and changes in brightness is tracked by the darkness metric. So hepatic organoids on the top panel there are not ready for passaging when they are less than 100 microns in diameter and display clear lumens. So I'm looking at the first image there, which is sort of 2 days post-seeding. As the culture matures, organoids take on a more regular round morphology, increase in size, and a few in the culture turn black. These cultures are now ready to passage, so I'm looking at the day 4 image there. Organoids have very much exceeded their passage window if the lumens of most of the organoids have darkened and many of them have collapsed, so this day 6 image. This is borne out in the time course, where at day 6, so that's 8 days post-seeding, you can see a marked increase in darkness in the darkness metric and an increase in the eccentricity metrics as collapsed organoids become less round. So for the intestinal organoids, as they mature, you observe an increase in the eccentricity metric, which corresponds to this very characteristic budding morphology that they take on. You also observe an increase in the darkness metric, which corresponds to accumulation of debris within the lumen of those objects. So in summary, these integrated metrics which kinetically quantify morphology features can be used to determine optimal times for passaging. And with that, I'd just like to summarize hopefully what I've demonstrated throughout this webinar, which is that the IncuSight image capture and analysis software is ideally suited to a range of 3D cultures. I've described a range of fully integrated applications, workflows, and automated life cell analysis solution for a range of 3D models, illustrated how real-time image-based approaches enables assessment and quantification of key biological phenotypes, and finally how life cell analysis approaches using complex advanced 3D models are ideally aligned to investigate cancer biology. Additionally, you can find more information on the areas that I've touched on today by following the links within this slide. So thank you for your attention.